Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this gluconeogenesis lecture, I will be going over important steps in gluconeogenesis process. Let's get into the details. Most of the steps of gluconeogenesis are simple reversal of glycolysis. So the only thing is the reversible reactions in glycolysis are the ones which will be using the same enzyme that was used in glycolysis. So that means the enzyme that was participating in glycolysis, that is conversion of glucose into pyruvate. So in all reversible reactions in glycolysis, all those enzymes, reversible reaction catalyzing enzymes that were there in the glycolysis will be used in gluconeogenesis to conduct the reaction in a reverse order, that is to come from pyruvate back into glucose. So the cell will use the same enzyme. That you need to know first. Now, what all those three irreversible reactions in glycolysis that needs a separate enzymes in gluconeogenesis. That's the another thing you need to know here to understand gluconeogenesis. Now the three irreversible steps in glycolysis which needs gluconeogenic separate enzyme, because the cell cannot use the same enzyme to conduct gluconeogenesis, because the enzymes which are catalyzing irreversible reactions in glycolysis, they cannot work in an opposite way, because they cannot take a product as a substrate and reverse it. Okay, so those irreversible reactions in glycolysis, they need a separate enzyme in gluconeogenesis. Now the glucokinase in the liver, which is converting glucose into glucose-6-phosphate, is an irreversible reaction. So in order to convert glucose-6-phosphate back into glucose, the reversal of the first reaction in glycolysis during gluconeogenesis, so there will be glucose-6-phosphatase enzyme, and this glucose-6-phosphatase is a gluconeogenic enzyme which will reverse a reaction catalyzed by glucokinase in the glycolysis. Similarly, for phosphofructokinase 1 enzyme in the glycolysis, which is the second irreversible reaction in glycolysis, so the corresponding enzyme is fructose 1,6-bisphosphatase. And the third irreversible reaction in glycolysis is done by pyruvate kinase enzyme, and the corresponding enzyme to reverse the reaction is pyruvate carboxylase and phosphoenol pyruvate carboxykinase enzyme. So like this, three irreversible reactions in glycolysis done by the first irreversible reaction in glycolysis is glucokinase, second is phosphofructokinase 1, third is pyruvate kinase. These three irreversible reactions are taken care by having a separate enzymes to conduct a reversal of these irreversible reaction in glycolysis, and those enzymes which are written correspondingly are glucose-6-phosphatase for glucokinase, fructose 1,6-bisphosphatase for phosphofructokinase 1, and pyruvate carboxylase and phosphoenol pyruvate carboxykinase for pyruvate kinase enzyme. You got to remember all of them, and that's how we can learn how gluconeogenesis can go on, how to reverse a pyruvate back into glucose. Now here in this figure, the same concept that we have seen in the previous slide is given in the form of a diagram here and the flow chart. Now let's first look at irreversible reactions in glycolysis and corresponding enzyme in gluconeogenesis, and also will take example of some of the gluconeogenic precursors to understand how these irreversible steps can be overcome by having a special enzymes in gluconeogenesis. Okay, so the first irreversible reaction in glycolysis catalyzed by glucokinase enzyme, which is converting glucose into glucose-6-phosphate, and the second irreversible reaction catalyzed by phosphofructokinase 1 catalyzing conversion of fructose 6-phosphate into fructose 1,6-bisphosphate, and the third irreversible reaction catalyzed by pyruvate kinase converting phosphoenol pyruvate into pyruvate. Other than these three irreversible reaction, rest of all the other reactions that is shown here, they are all reversible. So I'm going to reverse all of them just to make sure they are all reversible here. Okay, so that means all the enzymes that are conducting these reversible reactions can be used to convert pyruvate back into glucose. Now let's see the corresponding enzymes in gluconeogenesis for the irreversible reactions in glycolysis. So for glucokinase, we need to have a separate enzyme for gluconeogenesis, that is glucose-6-phosphatase enzyme. What this enzyme does? The glucose-6-phosphatase enzyme, it is going to convert glucose-6-phosphate into glucose. It's a reversal of the first reaction in glycolysis. We are going in an opposite direction now. For PFK1, phosphofructokinase 1, the corresponding enzyme is fructose 1,6-bisphosphate because it's going to break fructose 1,6-bisphosphate into fructose 6-phosphate and inorganic phosphate. Now, the pyruvate kinase reaction, the corresponding enzyme, there are two of them. One is pyruvate carboxylase, which will convert pyruvate into oxaloacetate, and then the other is phosphoenol pyruvate carboxykinase, which is going to convert oxaloacetate into phosphoenol pyruvate. So these two are the corresponding enzymes for pyruvate kinase enzyme. Like this, the corresponding enzymes for irreversible reactions in gluconeogenesis can be taken care so that there will be reversal of pyruvate all the way into glucose. Okay, this is the major thing for understanding of gluconeogenesis. So if you know how to reverse irreversible reactions in glycolysis, so that means you will understand for sure what is gluconeogenesis is. So you don't need to go make a special effort to go through our reversible reactions in glycolysis because same enzymes are used in gluconeogenesis to reverse in an opposite direction. Now let's take precursors for gluconeogenesis is a lactate. Lactate is a 3-carbon molecule, and alanine coming from skeletal muscle degradation, protein degradation. So alanine also can contribute 3-carbon going into gluconeogenesis. Now note that glucose is a 6-carbon molecule. So in order to make 6-carbon molecule, we need to get 2 3-carbon molecules. Either it can be 2 lactates or 2 alanine going into 1 glucose formation, or it can be 1 lactate and 1 alanine going together, like 1 lactate bringing 3 carbon, another alanine bringing 3 carbon. So 3 plus 3 carbon make 6-carbon glucose. How that is done? So lactate is converted into pyruvate, done by lactate dehydrogenase. We are going to see that. Once you get pyruvate, pyruvate has to be converted into oxaloacetate because pyruvate cannot go back into phosphoenol pyruvate because we don't have any enzyme which can revert that reaction. So that's why pyruvate will be converted into oxaloacetate, and that is done by pyruvate carboxylase reaction, and oxaloacetate converted to phosphoenol pyruvate done by phosphoenol pyruvate carboxykinase reaction, and the phosphoenol pyruvate go all the way to glyceraldehyde 3-phosphate. So that means we have got 3 carbons, glycolytic intermediate, glyceraldehyde 3-phosphate there. Now we need 3 more carbons. So for that, let's bring alanine. Alanine can be converted to pyruvate, pyruvate into oxaloacetate, oxaloacetate converted into phosphoenol pyruvate, and phosphoenol pyruvate going all the way to glyceraldehyde 3-phosphate. So that means we got 1 more molecule, so 3 more carbons. Now the glyceraldehyde 3-phosphate can be converted into dihydroxyacetone phosphate. So that is basically interconversion there. So we have just moved that 3 carbons other side. Now 3 carbon dihydroxyacetone phosphate, 3 carbon glyceraldehyde 3-phosphate, they both combine to make a 6 carbon fructose 1,6-bisphosphate. Now the fructose 1,6-bisphosphate will break into fructose 6-phosphate and inorganic phosphate done by fructose 1,6-bisphosphatase enzyme. So we still have the 6 carbon there. Now the fructose 6-phosphate converted to glucose 6-phosphate, that's done by phosphoexose isomerase, and now glucose 6-phosphate has 6 carbon there, and this glucose 6-phosphate will go into endoplasmic reticulum, where in the membrane of endoplasmic reticulum there will be an enzyme called glucose 6-phosphatase. It's going to remove phosphate at the 6th carbon of glucose, and there you get glucose, and that's how 6 carbon glucose can be made by using 1 lactate and 1 alanine. Or it can be done as 2 lactate going into reaction 2 times up to glycerol-D8 3-phosphate, and then both dihydroxyacetone phosphate and glycerol-D8 3-phosphate combine and make glucose. So that's another way of doing it. Or it can be another precursor that is shown here is glycerol. Glycerol is a 3-carbon molecule coming from adipose tissue triacyl glycerol lipolysis. Now the glycerol converted to glycerol 3-phosphate by glycerol kinase enzyme, not written here, and this glycerol 3-phosphate, I'll write it down, glycerol kinase, enzyme name is glycerol kinase, which will convert glycerol into glycerol 3-phosphate. Once you get glycerol 3-phosphate, it is converted into dihydroxyacetone phosphate done by glycerol 3-phosphate dehydrogenase. And once you get dihydroxyacetone phosphate, and this is a 3-carbon molecule, so 1 glycerol and 1 lactate make 1 glucose, or 1 glycerol, 1 alanine make 1 glucose, or 2 glycerol molecule making 2 dihydroxyacetone phosphate. In that, one of the dihydroxyacetone phosphate can be converted into glycerol-D8 3-phosphate, and they both combine to make fructose 1,6-bisphosphate, and all the way they can go into glucose. So this type of wagibility is also there. Okay, so like this, a gluconeogenic precursors can be converted back into glucose by reversal of most of the reactions in glycolysis except 3 reactions, and we have a corresponding enzymes to reverse those 3 irreversible reactions in glycolysis. Okay, this is the major thing that you must remember in order to understand gluconeogenesis process. Okay, so here let's divide 3 irreversible reactions in glycolysis that needs to be bypassed in gluconeogenesis. Next slide. So it can be studied as conversion of pyruvate into phosphoenol pyruvate, conversion of fructose 1,6-bisphosphate into fructose 6-phosphate, and conversion of glucose 6-phosphate into glucose. These are the 3 steps that we can study, because these are the reactions that needs a special enzyme to take a gluconeogenic precursor all the way back into glucose in gluconeogenesis process. Now lactate, which is one of the major precursor for gluconeogenesis, is converted into pyruvate by lactate dehydrogenase, where lactate is oxidized into pyruvate, and once you get pyruvate, so pyruvate can be reversed all the way back into glucose by a special enzymes involved in gluconeogenesis process. Similarly, alanine, which is another major precursor for gluconeogenesis, so it can be converted into pyruvate by transamination reaction, and the enzyme for transamination is alanine transaminase, and pyridoxal phosphate is required for this transamination reaction. So once you get pyruvate can be taken into gluconeogenesis, reversal of glycolysis, so all the way back into glucose. Of course, you need 2 pyruvates to do that. 2 alanine to 2 pyruvate, or 2 lactate into 2 pyruvate to make 1 glucose molecule. Okay, so that's how we can the cells initially convert alanine into pyruvate and lactate into pyruvate and take that pyruvate into gluconeogenesis process. Now let's see how to convert a pyruvate into phosphoenol pyruvate. So as I explained before in my previous slide, that conversion of pyruvate into phosphoenol pyruvate, it needs 2 gluconeogenic enzymes, which are special enzymes as we have seen, because these 2 enzymes are corresponding enzymes for pyruvate kinase irreversible reaction in glycolysis. Remember, pyruvate kinase enzyme, it is going to convert in glycolysis, it is going to convert phosphoenol pyruvate into pyruvate. So now what cell is trying to do here in the cell? So pyruvate has to be converted back into phosphoenol pyruvate. We cannot use same pyruvate kinase enzyme because phosphoenol pyruvate to pyruvate is done by pyruvate kinase enzyme, which is an irreversible reaction. We cannot use the same enzyme to reverse it, and also we don't have single enzyme to reverse pyruvate back into phosphoenol pyruvate. That's why we have two enzymes to do this. One is pyruvate carboxylase, other is phosphoenol pyruvate carboxykinase. These are the two corresponding enzymes for pyruvate kinase reaction to reverse it back into phosphoenol. So pyruvate back into phosphoenol pyruvate, we need two enzymes. Let's see that here. So here as it is shown, alanine going into pyruvate, we have already seen that. Lactate going into pyruvate, we have already seen that. Once we get pyruvate, pyruvate has to go down into the mitochondrial matrix to be converted into oxaloacetate. Okay, so why? Because pyruvate cannot go back into phosphoenol pyruvate by using pyruvate kinase enzyme, because it cannot go in a reverse order. And also we don't have any enzyme, a single enzyme which can take pyruvate back into phosphoenol pyruvate in the cytoplasm. So pyruvate has to go down into the matrix of mitochondria. So what happens there? Pyruvate in the matrix of mitochondria, it has two fates there. One of the fate of pyruvate is to go towards acetyl-CoA done by pyruvate dehydrogenase complex, there. Other fate is to go towards oxaloacetate formation. Now, on which side this will be going on? Note that gluconeogenesis is going on under fasting condition. So under fasting condition, glucagon levels in the blood will be elevated, and what glucagon does? Glucagon is going to stimulate hormone sensitive lipase in the adipose tissue, and it causes breakdown of adipose tissue triacylglycerol, and thereby free fatty acid floods into the blood, and these free fatty acids are taken up by the liver, and these free fatty acids are transported into the matrix of mitochondria, and in the matrix of mitochondria, as it is shown here, they will undergo a beta oxidation step, and the beta oxidation product is acetyl-CoA and NADH+. That means there will be plenty of acetyl-CoA that will be elevated in the matrix of mitochondria, and also there will be lot of NADH+ be present in the matrix of mitochondria. So what is the effect of this? So the elevated acetyl-CoA and elevated NADH+, they will have a negative effect on pyruvate dehydrogenase enzyme complex, thereby pyruvate conversion into acetyl-CoA is blocked, and that is what is shown here in the conversion of pyruvate into acetyl-CoA. So pyruvate is not largely going into acetyl-CoA because pyruvate dehydrogenase complex enzyme is kind of negatively modulated. So what will happen? Now the only way for pyruvate is to go towards oxaloacetate formation, and that is done by pyruvate carboxylase enzyme, and also importantly, the elevated acetyl-CoA in the mitochondria will have a positive effect on pyruvate carboxylase enzyme itself. So acetyl-CoA is a positive allosteric modulator of pyruvate carboxylase, which is a gluconeogenic enzyme. So pyruvate is now taken into oxaloacetate by pyruvate carboxylase, which needs biotin, and it does the carboxylation. Now oxaloacetate, it has two fate again. Oxaloacetate can be converted into malate. That's the major fate of oxaloacetate, because in the mitochondrial matrix we have plenty of NADH already coming from beta oxidation. Conversion of oxaloacetate into malate will use some of those NADH and release NAD+, and that NAD+ can go into beta oxidation again, because there is a need for NAD molecules there. So conversion of oxaloacetate into malate will give opportunity for a cell to regenerate NAD+ molecule. Now once you get malate from oxaloacetate, malate moves out of mitochondria as it is shown in the figure into the cytoplasm, and in the cytoplasm malate is converted back into oxaloacetate while NAD is reduced into NADH+, and oxaloacetate now will be converted into phosphoenol pyruvate, a 3-carbon molecule, by releasing carbon dioxide, and that needs an enzyme called phosphoenol pyruvate carboxykinase. During this reaction, a high energy molecule GTP will be undergoing dephosphorylation to make a GDP. Okay, so that means there is a need of GTP here, and this GTP need for phosphoenol pyruvate carboxykinase can be met by one of the reaction in TCA cycle, that is succinate thiokinase reaction. Okay, now this is how we have converted alanine lactate into pyruvate, and pyruvate all the way back into phosphoenol pyruvate in the cytoplasm. So we have 3-carbon phosphoenol pyruvate in the cytoplasm. Now this phosphoenol pyruvate in the cytoplasm can go down into pyruvate, as it is shown in the arrow there, done by pyruvate kinase enzyme, but that doesn't happen generally. Why? Because under fasting condition, there is a glucagon hormone which is released, and you know glucagon is going to phosphorylate pyruvate kinase by its through its protein kinase A mediated action, and the phosphorylated form of pyruvate kinase is an inactive form of an enzyme, and that is what is shown there, and thereby pyruvate kinase enzyme is kind of inactivated. That's why phosphoenol pyruvate cannot go down into pyruvate. So phosphoenol pyruvate has only way to go, and that is to go back towards glucose formation, and that is how pyruvate is converted into phosphoenol pyruvate and directed into glucose formation. Now let's see the conversion of other precursors for gluconeogenesis into phosphoenol pyruvate. Once I explain you that, then I will take phosphoenol pyruvate all the way towards glucose. I will explain that. So first let me finish explaining what are the other precursors for gluconeogenesis that can make phosphoenol pyruvate. It is a propionyl-CoA. You know propionyl-CoA, which is a 3-carbon molecule coming from variety of propionyl-CoA sources, which I have explained with a mnemonic that you must remember, and that is Bob needs vitamin M. This is the mnemonic that you can remember as sources of propionyl-CoA. So the propionyl-CoA, which is a 3-carbon molecule, as it is shown here in the figure, coming from various sources. It is already written there: oxidation of odd chain fatty acids, bile acid synthesis. The cholesterol that is written there is actually bile acid synthesis, oxidation of methionine, threonine, isoleucine and valine, and branched chain fatty acid oxidation missing here. Okay, so all of them give propionyl-CoA. And this propionyl-CoA is converted into methyl-malonyl-CoA by an enzyme called propionyl-CoA carboxylase. And since it is a carboxylase, so it is incorporating carbon dioxide for carboxylation process, and also there is a use of ATP here. So with that, 3-carbon propionyl-CoA is converted into 4-carbon methyl-malonyl-CoA. Now the 4-carbon methyl-malonyl-CoA will be converted into succinyl-CoA done by enzyme methyl-malonyl-CoA mutase, and that needs deoxyadenosyl-cobalamin as the coenzyme. Now once you get succinyl-CoA, which is a 4-carbon molecule, succinyl-CoA is converted to succinate, succinate into fumarate, fumarate into malate, and malate moves out of mitochondria into the cytoplasm, and in the cytoplasm malate is converted back into oxaloacetate, and oxaloacetate is converted back into phosphoenol pyruvate. Okay, let me write it down. So what I explained here is the succinyl-CoA, the arrow that is shown here is the expansion of the arrow that I am writing separately. Succinyl-CoA is converted to succinate, succinate is converted to fumarate, fumarate is converted to malate, and malate will move out of mitochondria into the cytoplasm, and in the cytoplasm malate is converted to oxaloacetate, and oxaloacetate is converted into phosphoenol pyruvate, and that's where you got phosphoenol pyruvate, and this phosphoenol pyruvate goes into glucose formation by additional reactions in gluconeogenesis. Now, before I move on to how phosphoenol pyruvate is converted back into glucose, let me explain you a clinical aspect with this pathway. It is what if there is a mutation in a gene coding for propionyl-CoA carboxylase? What if there is a decreased activity of propionyl-CoA carboxylase because of mutation in a gene or even deficiency of biotin? So that will give rise to increased accumulation of propionyl-CoA, and this propionyl-CoA eventually will be converted into propionate or propionic acid, propionate, and this propionate builds up in the blood and appear in the urine. That's why the disease is propionic acidemia, which can be a genetic condition because of mutation in a gene coding for propionyl-CoA carboxylase, or it can be secondary to biotin deficiency. Remember that. So other thing is, what if there is a decreased activity of methylmalonyl-CoA mutase enzyme, or if there is a deficiency of vitamin B12? So what happens? There will be accumulation of methylmalonyl-CoA at that time, and this accumulated methylmalonyl-CoA will be converted into methylmalonate, and methylmalonate is an acidic molecule, so it is going to be accumulated and appear in the urine, or accumulation causes methylmalonic acidemia. Okay, so this is how two diseases can be caused because of decreased activity or deficiency in the coenzyme for these enzymes. One is propionic acidemia, other is methylmalonic acidemia. Either it can be congenital or it can be acquired because of deficiency of either biotin or cobalamin in propionic acidemia or methylmalonic acidemia respectively. Now let's move on to see how to take phosphoenol pyruvate into fructose 1,6-bisphosphate. So the pathway that is shown here is the conversion of phosphoenol pyruvate into fructose 1,6-bisphosphate. Now the phosphoenol pyruvate is reversed into 2-phosphoglycerate by the same enzyme that was used in the glycolysis. 2-phosphoglycerate converted to 3-phosphoglycerate, 3-phosphoglycerate into 1,3-bisphosphoglycerate by using ATPs. Note that 2 molecules of lactate, 2 molecules of alanine going into 1 glucose molecule. So that means these reactions need to run 2 times to go towards the glucose. That means wherever ATPs are entering in, you need to count them as 2. Okay. So 3-phosphoglycerate going into 1,3-bisphosphoglycerate by phosphoglycerate kinase enzyme. 1,3-bisphosphoglycerate converted to glyceraldehyde 3-phosphate done by glyceraldehyde 3-phosphate dehydrogenase enzyme. So we got glyceraldehyde 3-phosphate there. Once you get glyceraldehyde 3-phosphate, if you are bringing 2 lactate in all the way here or 2 alanine all the way here, so you will have 2 glyceraldehyde 3-phosphate there. And out of these 2 glyceraldehyde 3-phosphate, one of it can be converted to dihydroxyacetone phosphate, and now both of them can condense to make fructose 1,6-bisphosphate, and that is done by aldolase B enzyme, reversible reaction in glycolysis. Now the fructose 1,6, that's how fructose 1,6-bisphosphate is made, from phosphoenol pyruvate. On the other side, from glycerol is converted to glycerol 3-phosphate by glycerol kinase enzyme, as it is shown there, by using ATPs. And glycerol 3-phosphate converted to dihydroxyacetone phosphate done by glycerol 3-phosphate dehydrogenase. So making dihydroxyacetone phosphate. So if you have 2 glycerol making 2 dihydroxyacetone phosphate, one of that will be converted into glyceraldehyde 3-phosphate. They both combine and make fructose 1,6-bisphosphate. That's how we made fructose 1,6-bisphosphate. So we have taken care of 2 hurdles there. One is to bring pyruvate into oxaloacetate by pyruvate carboxylase special enzyme, and oxaloacetate brought out into the cytoplasm and converted back into phosphoenol pyruvate by other enzyme, phosphoenol pyruvate carboxykinase. Both of them are corresponding enzymes for pyruvate kinase. So we made phosphoenol pyruvate there. And the phosphoenol pyruvate is taken all the way into fructose 1,6-bisphosphatase, where our next hurdle comes in. That is the second irreversible reaction in glycolysis done by PFK1 that has to be reversed now. So let's see how that happens. So I have already explained this particular reaction here, that is conversion of glycerol into glycerol 3-phosphate done by glycerol kinase enzyme using ATP, and glycerol 3-phosphate converted into dihydroxyacetone phosphate done by glycerol 3-phosphate dehydrogenase. So that's how we get dihydroxyacetone phosphate, which can combine with glycerol diet 3-phosphate to make fructose 1,6-bisphosphate. Now we are at fructose 1,6-bisphosphate. Let's see how to take it back. Now the fructose 1,6-bisphosphate is converted into fructose 6-phosphate. That is done by fructose 1,6-bisphosphatase enzyme. Let me write it down how that is made possible. So here is the fructose 1,6-BP, that is fructose 1,6-bisphosphate. So it is taken into fructose 6-phosphate. So after dephosphorylation of first carbon inorganic phosphate removed here, and that is done by fructose 1,6-bisphosphatase enzyme. It is simple dephosphorylation process, the phosphate attached to the first carbon. So this is how we make, we reverse fructose 1,6-bisphosphate back into fructose 6-phosphate. What if there is a deficiency of fructose 1,6-bisphosphatase genetically? What will happen? So that leads to accumulation of fructose 1,6-bisphosphate here. Note the term that there is 2 phosphates present in this molecule. All these 2 phosphates kind of trapped there. It is a trapping of inorganic phosphate in the form of fructose 1,6-bisphosphate, and this leads to decrease in inorganic phosphate pool in the cytoplasm. And whenever there is a decrease in the inorganic phosphate pool in the cytoplasm, it will lead to decrease in electron transport chain, it will lead to decrease in the TCA cycle, it will lead to decrease in the pyruvate dehydrogenase complex enzyme, it will lead to increase in the lactate formation, it will lead to fasting hypoglycemia. Why fasting hypoglycemia? It is because decrease in glycogen degradation, because glycogen degradation needs inorganic phosphate. Decrease in gluconeogenesis. Why decrease in gluconeogenesis? Because decrease in electron transport chain will lead to decrease in ATP synthesis, and when there is a decrease in the ATP synthesis, gluconeogenesis that needs ATP, it will decrease. So that will lead to fasting hypoglycemia. And since inorganic phosphate is kind of trapped here, there will be more ADPs which will break into AMPs. AMP further go into degradation to make uric acid. So patient will have hyperuricemia. Okay, and these things will lead to liver damage. It will lead to kidney damage. Patient will have liver failure, kidney failure, so many things, right? So this is how deficiency of fructose 1,6-bisphosphatase enzyme, which will lead to signs and symptoms very similar to hereditary fructose intolerance. Hereditary fructose intolerance. So I have already explained you hereditary fructose intolerance in the fructose and galactose metabolism. So you may refer to that video to understand all the metabolic effects of hereditary fructose intolerance. So whatever things that I have explained here briefly, so all of them are explained in the hereditary fructose intolerance. So differential diagnosis for hereditary fructose intolerance can be fructose 1,6-bisphosphatase enzyme deficiency. To a certain extent, even the signs and symptoms of classic galactosemia can also be correlated with fructose 1,6-bisphosphatase enzyme deficiency. Now let's look at what happens, how glucose 6-phosphate is converted into glucose. Because what we have seen so far from the previous slide is fructose 1,6-bisphosphate is converted to fructose 6-phosphate, and the fructose 6-phosphate will be converted into glucose 6-phosphate by phosphoglucoisomerase enzyme, and then glucose 6-phosphate has to be converted into glucose, and that's the third hurdle that we need to take care, because the first irreversible reaction in glycolysis is glucokinase catalyzed reaction. Now we need to reverse glucose 6-phosphate into glucose by a special enzyme, and that special enzyme is glucose 6-phosphatase enzyme, which is located in the membrane of endoplasmic reticulum. That means cytoplasmic glucose 6-phosphate, which is coming all the way from lactate or alanine or glycerol or propionyl-CoA all the way to glucose 6-phosphate in the cytoplasm, that has to now go into the endoplasmic reticular membrane. Now, in order to go there, in the endoplasmic reticular membrane, so we have a transporter called T1 transporter. Now, this T1 transporter, it is going to allow transport of glucose 6-phosphate to enter into the lumen of endoplasmic reticulum. Now, that is what is the role of T1 translocase or T1 transporter, glucose 6-phosphate transporter, also called as T1 transporter. So, it is going to allow glucose 6-phosphate to get in, and that's where glucose 6-phosphatase enzyme is attached there. And this glucose 6-phosphatase enzyme, it is going to convert glucose 6, so as it is shown here, this is the catalytic subunit of glucose 6-phosphatase. So, it is going to break glucose 6-phosphate into inorganic phosphate and glucose. Now, the inorganic phosphate will come out of endoplasmic reticulum into the cytoplasm via T2 transporter, and glucose will come out of endoplasmic reticulum into the cytoplasm via T3 transporter. Okay, so like this, glucose 6-phosphate, which is coming all the way from lactate, alanine, propionyl-CoA, glycerol all the way to glucose 6-phosphate in the cytoplasm in gluconeogenesis reaction, now has to go into the endoplasmic reticulum to be converted into glucose, and that glucose comes out of endoplasmic reticulum into the cytoplasm in the liver, and from the cytoplasm it is secreted out of the liver into the bloodstream. And that is what is the one that will maintain our blood glucose in the initial first 12 hours to 24 hours of fasting and starvation. And this is how gluconeogenesis process will go on. So, defect in glucose 6-phosphatase enzyme, it will give rise to a disease like glycogen storage disease 1A. Defect in a T1 transporter, it will give rise to glycogen storage disease 1B. So, we are going to study details of glycogen storage disease 1A and 1B later in glycogen storage disorders. Otherwise, make sure to remember how exactly the cytoplasmic glucose 6-phosphate is entering into the endoplasmic reticulum to be converted into glucose 6-phosphate, because the enzyme glucose 6-phosphatase is located there in the membrane of endoplasmic reticulum. So, that's all about how a gluconeogenesis process will go on in reversing alanine, lactate, propionyl-CoA, glycerol all the way back into glucose by using a reversal reaction in glycolysis, and also using a special enzymes that can reverse irreversible reactions in glycolysis. So, that's about important steps in gluconeogenesis. I hope this video has helped you in understanding that very important concept. Thank you.